my name is Natalie Shink, and I am an undergraduate student in the biology program at CSU Channel Islands. So I work in Dr. Harris's neuroscience lab, where we aim to characterize how genetics of an organism changes how their brain behaves in a multisensory behavior. So when we think of the human senses, we think eyesight, hearing, taste, touch, smell, and our orientation in space. But we're capable of sensing much more that helps us understand and perceive the world around us. Not only do we have all of these senses, we're able to sense things at the same time. For example, when we eat a cheeseburger, we're taking into the look of it, the smell of it, how it feels, and how it tastes. Despite appreciating this, we don't know the mechanisms behind how we process all this information, and that was where our research comes in, between repulsion and attraction. Our everyday life is full of such decisions that greatly affect our daily life, and how we commit is a complex process that is not understood. We have many pathways in the brain, like neurotransmitters such as serotonin and dopamine, that allow for decision making, but we are still far from being able to comprehend the pathway from decision to action. My research explores neuropeptides, which are small peptides used to communicate with each other that influence brain activity. Different neuropeptides are involved in a wide range of functions like mood, decisions, choice, and behavioral state. So we're exploring the exact mechanisms of how neuropeptides allow sensory information to be converted into decision-making behaviors that are still not understood. Neuropeptides like oxytocin, vasopressin, insulin, and neuropeptide Y are neuropeptides important for social interaction, appetite, mood, and decisions, and sensory processes in the brain. And we basically know nothing what is responsible for the behavior. And how do these neuropeptides regulate in a multisensory behavior? There are a variety of neurological disorders like Parkinson's disease, anxiety, and schizophrenia, and neuropeptides play a huge role in how these pathways regulate decision making. And a lot of these disorders are associated with processing multisensory behavior. So to study multisensory behavior, we use C. elegans. C. elegans are microscopic worms, and we use these worms to understand how the neurotransmitter system allows the brain to talk. The worm's brain is able to distinguish between different sensations that come from odors that may be attractive or repulsive to coordinate a decision. So C. elegans encode genes that share significant homology with mammalian systems like oxytocin or insulin. Their specific neuropeptides and receptors drive functions like mood, appetite, sensory perception, and decision making. And mutants are available for nearly all the nervous system expressed genes in the worm. So we examine mutant worms that lack individual neuronally expressed genes. So to assess multisensory behavior and decision making, we use this behavioral paradigm where we place the worms on a patch of food that they love, then we put a chemical right next to the food patch called known that they hate. They're taking in cues from both the food and the chemical at the same time. So we want to know how long it will take for the worms to make the decision to leave the food patch. This graph shows data from a wild-type worm. On the x-axis is time in minutes, and the y-axis is the average number of worms that left the food. So over time, it shows that the worms leave in the presence of the food and the chemical. And usually between 10 and 20 minutes, most of the worms have made the decision to leave the food. So in this first picture shows the start of the behavioral experiment, where all the worms are on the food, and after about 5 minutes, most of the worms have made the decision to leave the food, or they're nearly at the edge of the food. At 45 minutes, nearly all the worms have left the food except for a couple. So we want to examine mutants that lack neuropeptides and neuropeptide receptors to identify the role for neuropeptide signaling in known dependent food leaving. Many neuropeptide genes are not required for known dependent food leaving, and this is suggesting no involvement. And we've tested multiple mutants. So in these graphs, the black line indicates the wild type worm, and the blue and green lines indicate the mutant worms. Their leaving behavior is very similar to our wild type worm, and we put this data into a student t-test, and it shows no statistical difference. Something interesting is that we identified a neuropeptide receptor mutant that is defective in known-dependent food leaving. And in this graph, the black line shows the normal worm, and the red line shows the mutant worm. And it's leaving at a much slower rate compared to the normal worm. These pictures show at the 15-minute time point, and on the left shows the wild type worms at 15 minutes, and nearly most of them have left the food patch except for a couple. And on the right is our neuropeptide receptor mutant, and most of those worms are still in the food patch and haven't made the decision to leave. So we repeated this behavioral assay with the chemical benzaldehyde to determine the phenotypes are general to multiple types of repellents. And there is a huge statistical difference in their leaving rate. As you can see, the mutant is a red line, and they left at a much slower rate compared to the wild type worm. In conclusion, I performed a multisensory behavior assay to assess worm behavior of wild type worms and mutant worms. Many neuropeptides not involved, suggesting specificity in neuropeptide signals used. So we found that neuropeptide receptor 25 is involved in this multisensory behavior, and I want to know where in the nervous system that it plays a role. So my future goals are to investigate the mechanisms underlying this neuropeptide receptor in multisensory behavior. So how does this neuropeptide receptor regulate food repellent sensing circuit? So this neuropeptide receptor could play a role in the sensory neurons or the interneurons or the motor neurons. So something interesting is that this neuropeptide receptor is similar to the human somatostatin and chemokine receptor, which is involved in neurological disorders. This is suggesting an avenue to study types of receptors in complex decision-making. I would like to acknowledge the research students, faculty members, Dr. Sean Kelly, Dr. Sanchez, Betty Ortiz, and Dr. Lee for helping me with my presentation. I would also like to thank LSAM for funding my research, National Science Foundation, and the CSUCI for providing the worms. And a special thanks to the principal investigator, Dr. Harris. Thank you so much for watching my presentation, and I'm looking forward to the question session.